Had all these challenges now with the scale up. Um, so a typical scale up pattern for a mammalian cell line is, so you take your frozen cell line vial. What you end up having to do to get up to your production scale, let's say 20,000 liter facility, is you have to do a lot of intermediate scales, because mammalian cells don't really like to be by themselves. If you inoculate a huge tank with just a little bit of cells, mammalian cells aren't happy. They don't like that. They also don't like being too crowded. So you have to keep in this finite range of cell density for them to be okay. So that can be tough, and there are a lot of challenges associated with that portion. Now, the flip side is that most mammalian cells, well, all mammalian cells don't have a cell wall, right? So it's a little bit easier to handle cells like that to get your products out. And the better thing is, all protein products that we make in mammalian cell lines are secreted, or at least we engineer them to be secreted. The thing is, you don't have to bust open your cells to get at your product. Your product has already been secreted, so that purification step and some of those downstream steps end up being a little bit easier actually with mammalian cell culture. Just for the fact that if it's secreted, you don't have to now bust open cells to get at your product and then deal with purifying all of the cell debris and everything else away. There are various cell lines that are used to make all different types of products. Like I said, we started out really using bacterial cell lines like E. coli. We had a lot of expertise in that. The problem is there were a lot of limitations in terms of making mammalian protein products. They couldn't do some of the things like proper folding. They couldn't do a lot of the post-translational modification. So the industry moved towards using mammalian cell lines. So, for example, CHO cells have been the bread and butter of the industry. I mean, those have been used for 20, 30 years now. Because of the fact that they do have similar glycosylation patterns as humans. So that's one of the benefits. But anytime you are trying to make, a protein, a human protein in another species cell lines, you're always going to have challenges. And these are actually a lot of what engineers and scientists are doing at these pharmaceutical companies, are hitting every single point in the process and trying to make improvements to that. One of the things that that the FDA has to do is it's got a balancing act. It's got to protect the citizens without preventing, potential drugs and therapeutics from that could harm people. But I think the thing to always remember is a drug is a drug. There are side effects to every single drug that you will take. There is no such thing as a harmless drug. Any drug you take has a potential to do some harm. Now the FDA has to do that balance. The recent trend in the FDA is they're becoming very stringent. They're actually starting to, it's hard to say for sure. You never get to actually see all the clinical data. But the general trend seems to be a shifting towards really having to have higher and higher safety standards. Now, if you could say it's because of the backlash of things like Vioxx or some of these other drugs that have come out and have harmed people, but any drug is going to have the potential to harm a person. So it's always, is the benefit larger than that potential harm? One of the big issues that I think is very interesting regulatory-wise right now is the approval process for biologics that are, what's the word? That are basically the copycat drugs, right? The second generations. If you were going to call, we have generic antibiotics that come in, or generic drugs. So the big name brand comes through, they charge you, a lot more money for those, and then once those come off patent, then a slew of generics come around. Well, when you have a small molecule, it's pretty straightforward. You have a chemical formula, the structure, you can do NMR, whatever, to say, okay, this is my drug. Now, for a protein product, it's obviously not the case, right? The process defines your product. Um, for example, let's say you have an antibody that has the perfect amino acid sequence, but the glycosylation is different. Is that the same drug or is that something different? So some of these sort of generic biologics, that's a big issue right now of should the FDA have an accelerated process? So normally when you have let's say you are taking Lipitor, something like that, Lipitor, the cholesterol-lowering drug, and when that comes off patent, when the generic comes in, they usually have these abbreviated clinical trials, so you don't have to do the full extended studies that the people who made Lipitor did. They got to, if you're a generic company, you are basically able to do a lot of abbreviated studies because the studies have already been done on your product, and you can say, I have the exact same product. With a protein pharmaceutical, it's not that case. And so there's a big argument. Some people are saying that by not providing an accelerated track, that we are sort of preventing people from having access to drugs that they can't afford, right? So the first company that gets that new drug, they spent all that money on the R&D, they spent all that money on the clinical trials, so they get to price it higher. Now, if the generic comes in, and let's say we have an accelerated process for them to get approval, then now people have access to that same drug at a lower cost. The issue is, some people view this as, okay, these big, wealthy pharmaceutical companies don't want us to cut into their profits, so they're trying to say that, generic biologics are too different, that you can't do it. That's one side of the argument. The other side of the argument is they are the same thing. You are preventing access for people who have lower incomes. The cool thing that everyone talks about is personalized medicine, and I think most people believe that that is not a if it will happen, but just kind of when it will happen. Where, and you guys may have talked about some of these where you do more genetic screening to say, one of the things that's scary with taking a drug is I could give you the same amount as I give me, and it could have deadly consequences to me, but not to you, and we don't know why. Now, maybe with some genetic screening, maybe we could find out everyone who had really negative responses to that drug, maybe there's some underlying genetic reason to that. And if we could screen everybody, and before giving them that drug, to screen them and say, for your genetic makeup, this drug isn't going to work, or we know this other drug will work better. So when you have a headache, maybe there's going to be like some personalized version of Advil that works better for you, and then I have some, Motrin that works better for me. So, and that is huge because, the amount of sort of safety implications right there alone, just safety, is huge because now we might be able to prevent, when you start getting to these large clinical trials where you're testing thousands and thousands of people and you do have 1 or 2 deaths, maybe we could prevent those. We think about sort of development in terms of just the pharmaceutical industry, but development in other industries also parallels that. So let's say in the sequencing area, one of the big efforts is to figure out how to sequence a human genome for $1,000, right? If you can actually sequence everybody's genome for $1,000, you could then start to look more at these personalized medicines. So, development of technology all around, and that has to do with also maybe, how fast we can simulate protein folding. So maybe computationally, if we make advances over here in protein folding simulations, that will impact us being able to design drugs. So it's pretty amazing how advances in all sorts of technologies around us are going to help the biotech industry too.